Today, we're diving into a story about a scientific breakthrough that's tackling a huge invisible threat in our food supply. It's a story about a really big problem on our farms and a surprisingly small, pocket-sized solution. So, just imagine for a second, you're a farmer. Your animals are starting to get sick, but you have no idea why or how fast it's spreading. And every single day you wait for an answer, the problem just gets worse and worse. This is the reality for so many farmers, and it brings up a really tough question. How do you fight an enemy you can't even see before it's way too late? Well, the villain in our story today is a tiny bacterium called Mycoplasma synoviae, and trust me, it is a huge problem for the poultry industry, causing a sickness that just spreads silently and devastatingly through an entire flock. Now, this isn't just some chicken cold. We're talking about painful joint swelling and serious breathing problems. Birds that get infected, they stop eating, they don't grow properly, and their egg production just plummets. It spreads like wildfire, and what's worse, it causes severe immunosuppression. Basically, it just wrecks their immune system. That makes them sitting ducks for all sorts of other diseases. So you can see it's a massive issue for animal welfare and a huge economic hit for farmers. Okay, so if this disease is so bad, why can't we just test for it and stop it in its tracks? Well, that's where the race against time really begins. And for a long, long time, we've been losing that race. And here is the massive gap farmers have been dealing with. The so-called gold standard test can take up to 28 days to get a result. I mean, 28 days. A farmer's entire operation could be wiped out in that time. Yeah, there are some faster methods, but they need specialized labs, expensive equipment, and experts you just don't have sitting around on a farm. What's desperately needed is something fast, portable, and simple enough for a vet to use right there in the field. And that, right there, brings us to the hero of our story. Researchers have developed a brand new tool that's designed to basically shrink an entire lab down and put its power directly into the hands of people on the farm. It's called Insulated Isothermal PCR, or iPCR for short. Now, the easiest way to think about this is like a super specific genetic photocopier. It hones in on the one unique genetic fingerprint of the MS bacteria in a sample, and then in minutes makes millions and millions of copies. It turns a tiny invisible clue into this massive, unmissable signal, and it does it all inside a little portable box. And the process itself? It's incredibly simple. You just take a swab from a chicken, you pop it into this handheld analyzer, and you press a button. The machine does all the work, and in less than an hour, you get a clear, easy-to-read yes or no result. No sending samples to a lab, no waiting for weeks, just a straight answer right then and there. Okay, so this sounds amazing, right? But a cool new invention is one thing. Proving it actually works when it counts is something else entirely. The researchers had to put this new pocket detective through some really tough trials to see if it was really up to the job. You know, for any new diagnostic test to be trusted, it really has to clear three fundamental hurdles. First, is it specific? Does it only find the one thing it's looking for? Second, is it sensitive? Can it find that target even in tiny amounts? And third, is it accurate? Do its results match up with the old, reliable methods? Let's see how it did. First up, specificity. You have to be sure the test isn't going to raise a false alarm, and the results here were just perfect. You can see a clear, strong positive signal for the target MS bacteria, marked with a P. But for the 6 other common germs they tested against, nothing, not a peep. That's a perfect score. It proves the test isn't easily fooled. It only zeroes in on the one culprit it's supposed to find. Next up, sensitivity. Or, can it find a needle in a haystack? To figure this out, the researchers took a sample and just kept diluting it over and over, making it weaker and weaker. And look at this. In lanes 1 all the way through 9, the test still got a positive result, even at incredibly low concentrations. This is huge because it means the test can spot the disease in the very earliest stages of an infection when the bacteria is just starting to spread. And finally, the ultimate test. How does it do in the real world? They took 50 samples from chickens on actual farms and ran them through the new rapid test. It found 18 positive cases. Then they ran those same 50 samples through the slow, traditional lab-based test. The result? It also found exactly 18. This is exactly what you want to see. You get the same reliable answer, but in a couple of hours instead of a few weeks. So, what does this all add up to?
Well, a fast, sensitive, and accurate test is more than just a cool piece of science. It is a genuine game changer for how we protect our animals and, by extension, our food supply.
Let's just pause on this for a second. The entire process, from swabbing a chicken to getting a final actionable result, can be done in under 2 hours. A process that used to take the better part of a month can now be done in less time than it takes to watch a movie. That's not just an improvement, that's a revolution. And that speed, it changes absolutely everything. Farmers can immediately quarantine sick birds before a small problem becomes a full-blown outbreak. They can use the right treatments right away instead of just guessing. All of this means healthier animals and less suffering, and it provides the kind of economic stability that protects a farmer's livelihood and keeps our food system secure. So yeah, this technology is a powerful new weapon against one specific disease. But the idea behind it, that's so much bigger. It proves that we can pull complex, life-saving diagnostics out of the sterile lab and put them right where they're needed most, out in the real world. And that just leaves you wondering, if we can successfully bring the lab to the farm, what other urgent problem can science solve next?